Okay, so thank you very much, and thanks for the invitation to this school. I am not really, I would say, an expert of stochastic analysis, but I think it's very good to mix fields. My expertise is more in PDEs and modeling, and but I'm say on a daily basis confronted to stochastic models, and then we'll see from these examples I'm going to give now. So roughly speaking, my course will be divided into 3 parts, roughly speaking 1 hour each. So this lecture will be, mostly first part and the start of the second part. So in the first part, it will be more a kind of an entertainment. I will give you a certain number of examples of problems where biological problems that arise and that require a mathematical understanding. So that's the first part, and the second and third part will be more mathematical, more into the the core of the subject, and the hard core of the matter. So one is will be about coarse graining, so the passage from micro to macro, which is, I think, one of the big topics of this meeting, but I'll provide a slightly different perspective, more PD oriented. And the last part will be about phase transitions. Okay, and as an example, I will treat mostly the Vicsek model, and I will tell you more about this model in the course of the lecture. So the first part will be about essentially giving you a broad perspective of and examples which are actually taken for my own experience, but I think that they can be interesting for a general perspective. Okay, so the first thing I'd like to ask, because I'm interested in collective dynamics, and what does collective dynamics mean, or what does it imply? And I'm going to give you a couple of examples. So the first thing is this experiment that I was associated to, which deal with sperm. So this is a sheep sperm, and this is a channel which is engraved in a kind of a plastic material. So you have to think of this is about 2 millimeters, and this is about 200 micrometers. And so what's happening is that the sperm is undiluted. So for sheep sperm, it's a highly concentrated suspension of active particles. So the sperm move, and they are basically at close packing. So when they move, they have, in some sense, to decide to move in the same direction. Otherwise, they are going to block each other, right? And so if you confine the sperm into this narrow channel, then in some sense, then the sperm have to decide collectively to turn either clockwise or counterclockwise, and in this case, they have decided to turn clockwise, right? So there is a coordination of the particles, and here you see that sperm is not that, is basically just a engine that extract energy and moves, right? So it's not very smart. Maybe what's inside is maybe smart, but not the thing itself. But in spite of this, then there is a collective decision that's made. And here you have an example of a symmetry breaking, because you have a decision to turn in this direction. However, the decision could have been to turn in the other direction. Right? And actually, so why doing these experiments? This is because this collective motion of sperm is an indicator of fertility. Okay, so there are diagnostics that's used by people in artificial insemination centers to actually select the good sperm, that is the sperm that will be used for commercialization. And in this project, which was funded by the French Agence Nationale pour la Recherche, was to provide some objective measurements of that collective, the ability of the sperm to move collectively. And this is one of the solutions that we propose. We patented it, but for some reason, the patent was abandoned. Anyway, so yeah, this is actually a paper which just appeared in the Journal of Royal Society Interface, which is open access, and you can actually download some videos where, in some instances, actually, which is quite interesting, is that you may have reversals of motion. So for some very active samples, you have the sperm spinning in one direction and then decide to spin in the other direction and oscillates between left and right. So, so that's the reason why you have this kind of spinning of a change of direction is not really understood yet. Okay, so this is one example of collective dynamics coordination. So let me give you another example, and another example is self-organization. So there is absolutely no correlation between the annular geometry of the previous experiment and this one. These are pedestrians. These were experiments conducted in University of Rennes in Brittany, and these are students hired for the occasion. And so in this experiment, the position of the pedestrians were recorded. And so you see that you have, you have spontaneous lane formation. Actually, we have recorded data, and actually in this paper in particular, tried to provide statistical evidence of this lane formation, right? So this is essentially due to the fact that motion is easier when you follow the one who is in front of you, right? So that's a kind of a natural way to do, and that's spontaneously formed lanes. However, you can see that the lane, break up at some points because of some people behaving differently than others, or things like that. So self-organization is another feature of collective dynamics. Another thing that is another feature, maybe there are many more, but it's just the ones that I like to highlight here, is pattern formation. So here is a different story, is actually a tissue, adipose tissue of a mouse. So there is no movie here because to acquire this image, you have to kill the mouse, so you can't really do it, a movie. So you have to fix the tissue and shine a light on this, and then you have So these red spheres are adipocytes, so there are cells full of fat. So there's basically balls of fat, and they are grouped into these structures, and then around the structures you have a network of fibers which is made of collagen. And so the question is, this kind of structure is very common in many tissues, right? So if you take the liver, if you take pancreas, if you take of course the fat, if you take many tissues, you have muscles, you have a formation of groupments of cells, and they are grouped into clusters that are surrounded by fibers. So there must be some generic phenomenon that take you to these structures. And so the emergence of patterns like this is another feature of collective dynamics, because in the formation of the tissue during embryogenesis, you have cells that move, and they move collectively, and eventually you get this kind of structure, right? So cell collective migration is actually a very important topic both in embryogenesis and also for diseases like cancer, for instance, understanding how cancer metastases and things like that. This is an issue of collective cell migration, right? So this is the kind of things that, for me, characterizes collective dynamics, and for which we would like to have mathematical tools to analyze. And by the way, if you have any question, please feel free to interrupt me. This is, I wish you, there is a kind of interaction with you. So these are examples. And so all these, what's important in these structures that appear is that they are not really encoded into the individual's interactions. Like, for instance, in the lane formation, the pedestrians, they move, they do not think of really what they're doing. Maybe they are thinking of, you know, something else, and then they act really spontaneously. So there is no really encoding of the you know, of this big structure into the individual behavior, and yet these structures emerge at the large scale. So we're speaking of an emergent behavior, where you have large-scale structures that appear that are not encoded into the individual particle, or not directly encoded into the individual particle interactions. This is also something that's sometimes called a complex system. Although complex system doesn't mean much, you know, nobody is going to advertise the fact that he's doing simple systems, right? It's not very much. You can't really get a lot of grant money from simple systems, right? So it's better to speak of complex systems. Right, so here is an example, actually a toy example, and it's when I say toy, I mean it, because we are, you know, it's actually toys that I went to the La Grande Récréation, basically the French Toys R Us, with a purchase order from CNRS, asking for 100 of these toys. Okay, I don't tell you how much effort I had to make into getting this purchase order in the first place, and the number of explanations I had to give. In any case, the guy who sold me this was very happy because it was the first time he was working with CNRS, right? He was usually working with the local, elementary schools, okay? So he said, okay, maybe there was a new market. He was very interested. He told me that, you know, I could come back anytime. He would be very happy. Okay, we didn't really come back so far, but what we did basically, we were trying to see if we could see an emergent behavior when you put a lot of these little things. So these are called bristle bots. So you see these are small bristles, small, legs. And, there's an engine, a small battery and an engine. The thing vibrates. And the vibration, because of the bristles, actually it sets the thing into motion. And this is what you get. And so we did serious work on that. Actually, we did, we recorded videos and tracking and stuff. And the reason for that is what originally we thought we would get maybe a model for a sperm experiment. The problem with the sperm is that it's very difficult to track individual cells from the images I showed before because they are so much close to packing that, when you follow one individual cell, maybe it's hidden by another one, or it just goes away from the focal plane of the microscope. So we said, okay, maybe this system would be a nice model for our sperm. And unfortunately, it didn't do what we expected, but it did something else. It did clogging on the boundary like this. And so we decided to study this. And so this is another example of an emergent behavior. And actually, it shows also some symmetry breaking, because from a very completely isotropic state, you get these clogged states. And so we are still working on, trying to model these coagulation of bristle bots on the boundary, and trying to, match the model with these data. So this means that collective behavior and emergent behavior is not something that you only find in the living systems. You have many examples in the inert matter that also shows this kind of emergent behavior. You can think of, spin systems also as doing these kind of things. Although in most of the cases in physics, you have an energy driving these phase transitions. And it's very difficult, actually, and almost impossible in many cases to actually highlight a kind of a Hamiltonian structure behind these systems. So you have, in some sense, to leave a little bit this world and to invent different paradigms. So just one of the things that make these systems interesting but also difficult is actually precisely the emergence of phase transitions, right? So, for instance, the emergence of order from disorder. So here, typically, I have a system, so that's one of my favorite systems was proposed by this physicist, Tamás Vicsek from Hungary, in a actually quite celebrated paper from '95, jointly, by the way, with the Israeli team of Eshel Ben Jacob, who unfortunately passed away a couple of years ago. And so he invented a very simple model in which, and we will come back later on this, where particles just move with constant speed and try to align with the neighbors, but subject to some noise, and according to whether you have large or small noise, then you will have either disorder or order. And here in this video, which has stopped, unfortunately, I don't know why. How can I get back to work? Okay, let me try. Okay. Okay, so I guess the only way is I quit the presentation and I go here. Yeah. And yes, so here you have a simulation of a Vicsek model, and this is actually to highlight the emergence of a phase transition. There is a very small difference in the amount of noise in between the two simulations, but you see in the smaller noise case, you have formation of clusters with a ordered motion, so a specific direction of motion, whereas in this case, you still have a completely disordered medium, right? So, and this transition occurs in a very narrow region of parameter space, in this case, the amount of noise. It could be also the density, okay? So you have transition to order when you increase the density, which is, if you think, a little bit paradoxical, because increasing usually in normal systems like gases, when you increase the density, you increase the temperature, so you increase the disorder, whereas here, when you increase the density, you increase the order. So you have a kind of, say, negative temperature effect in these systems. And in any case, as I said, the degree of alignment, what we call the order parameter, changes drastically when you vary the noise in a very narrow region of parameters, and that's actually signature of a phase transition, or bifurcation, if you like. And there are many examples actually of different classes of phase transition you can see in these systems. One of the examples is symmetry breaking, as we already saw with the sperm, with the with this Vicsek model, where you're breaking the isotropic, the rotational invariance. So typically here is an experiment with fish, real fish, which was conducted by my friend from Toulouse, Guy Terolas. And here is put some fish in a tank. These were experiments conducted in the French island La Réunion. So you see we are having really a tough job. We have to go to La Réunion to do these kind of experiments. So you see that, okay, the video has stopped, but you could see that with a small number of fish, you had a ordered motion, a collective motion of this group of 5 fish, whereas in this case of a bigger group, then there is no preferred direction of motion. So this is an example of a symmetry breaking phase transition. Incidentally, it occurs in a different, actually completely different order as the previous one. This is here, the phase transition occurs when you decrease the density, a little bit more like what you expect to see in gases. However, here it's expected that this is really the influence of the boundary and the confinement in the tank is actually playing a very important role. Other examples of phase transitions are what I would call packing. So these are videos again taken by Terolas the Parc du Mercantour in the southeast of France, and so they put the camera on some of it at top of the hill, and they were looking at these herds of sheep. Here you have a dog that you may see actually running here that provides a kind of a confinement force that sort of confines the sheep into a kind of incompressible medium. Whereas here it's really, you have a lot of space. So here you have basically a compressible phase, you have space in between the particles, whereas here you have a typical example of an almost nearly incompressible phase. So you have transitions between compressible to incompressible through through volume exclusion constraints. So the volume exclusion constraint plays a very important role, because most of the case when you deal with sheep, with cells and so on, you can't just suppose that the particles are point particles. They have a certain extension, okay? So there is a volume exclusion. And another example of a phase transition that I actually think is very important is a transition from a continuum to networks. So here you have an experiment, again conducted by a student of Guy Terola's in Simon Garnier, who's now in the US, with actual ants. And you have, here you have a hole, and you have an arena disk, and the ants are released by the hole, and then they start to wander around. So at the beginning you have kind of like a diffusion, standard diffusion, but there is as you probably know, the ants release a chemical, a pheromone, and then the further individuals try to follow these chemical trails, right? And this produces a reinforcement of the trails. And here we have simulated this phenomenon using a model where in blue you have the ants that are moving, released by the center, and in green you have the pheromone trails, and you see that you have this phenomenon of trail reinforcement, which produces this concentration on a network. And so according to the parameters that you put in the model, you can have different network characteristics. So for instance, the trails may be a bit wider or a bit narrower, or there may be more or less connections between or branches and things like that. So here again, the emergence of a network is not directly encoded into the behavior of the particles, but they emerge from it in a very indirect way. So if you want to characterize the network characteristics from the parameters of the model, you really have a lot of work, because you don't know a priori why this given set of parameters gives rise to this particular structure. You really have to explore the parameter space and trying to really do a huge statistical work in order to understand, or at least give a classification of the patterns that you get. So that's the question for when you have emergent behavior. This connection between the individual behavior and the big large-scale pattern is not easy, right? And you need some tools to do that. And one of the tools we are going to use is coarse graining. Okay, coarse graining gives you allows you to go from the individual-based model to a large-scale model in which you maybe, if you're lucky, be able to analyze the large-scale pattern in a better way than using with the individual-based model, right? So this is one of the things that we are trying to do. Yeah, so about coarse graining, precisely what's the methodology. Basically, we are doing what we've seen already in this course. So there are essentially two steps. So the first one is that we are trying to get what we call the mean field model, or the mean field kinetic equation. So essentially, this is basically a probabilistic description or statistical mechanics description of the ensemble of agents. So here I've imagined that I have a box with just Newtonian particles with positions and velocities. In practice, it can be more complicated and have more variables like the size, if you have a cell, or if you're dealing with elongated particles, that could be also the elongation of the particles. But in any case, for this case, we have positions and velocities, so we have a system of interacting particles, and rather than looking at individual equations for each of the particles, we are trying to get a statistical description through this probability distribution function of particles at position x and velocity v and time t. So of course, because the particles are interacting, it's not clear whether you can actually describe the whole system with a single distribution function. That means just looking at a representative particles. If you have correlations between the particles, you may have to take into account the 2 or 3 or more particle correlations. So the ability to write an equation for just a single particle requires the influence of any given particle on the system be very small, what we call the propagation of chaos, and what we've already seen several times during this course. So one of the features of the collective dynamic system is that propagation of chaos may be untrue. And so we have been able to actually exhibit. So there was a question that was actually raised by Paolo yesterday, which was the question of whether, so obviously this ability, the fact that the influence on a given particle on the whole system is very small is likely to be true only in the limit of the large number of particles, n to the infinity, right? So usually what you do is, okay, you take a system of particles, imagine that you have a large number of particles at n to infinity, and prove, if you're lucky, or assume, if you are lazy, that I rather prefer the second option, actually. You can lump the description of the system into just this single particle distribution function. So this is basically, this step requires n to infinity. However, we're interested very often in the stationary states, which means letting t to infinity. And then there's a question whether we can actually invert the limit, because when we are able to pass from here to here, we let n to infinity, but usually this is only true on a finite, any but finite interval of time. All right? There is actually a very, I think that one of the first papers or personal work where this was really highlighted is the work of Cox on the what we call the Cox dynamics, right? So where it's really, you really see that you trade off, in some sense, the uniformity in T that you would get if you solve the equations. When you solve the equation at this level, usually if you're if the system has good properties, you can solve it for any t and uniformly in t. But in some sense, in order to get the limit n to infinity, you have to trade off this uniformity in time to get the uniformity in n, in the number of particles, right? And so this is where you lose the uniformity in t, and you have a finite, only a finite time validity of this limit. And then once you get there, you're looking at t to infinity, and then, of course, you can wonder whether you're doing well, whether the stationary states of this guy have anything to do with the stationary states of this guy. And that's actually what happens here. What we've shown is that our system, I'm not going to give the details because I've decided to focus on a different thing, but basically what's happening in this system that we have looked at in this paper, we have exactly this. We have propagation of chaos in finite time, but we are able to show that if you look at much longer times, which are what we call hydrodynamical times, then the propagation of chaos is not true anymore, and we have an explicit counterexample, right? So this is something that you need to bear in mind when you are looking at this question, is that you may face problems with the uniformity of your propagation of chaos in time, right? Now, for gas dynamics, it's not proved uniformly in time. Actually, the proof by the Boltzmann, the hard sphere dynamics goes to the Boltzmann equation by Landford is actually only valid for a short time. However, it turns out by other statistical properties that the chaotic equilibrium is a solution of the is a stationary solution of the dynamics. So you, in some sense, it's behaving fine for a stationary state. So you can hope that at someday you will be able to fill the gap between so finite time to t to infinity for usual gas dynamics. But for these systems which exhibit collective behavior, this is hopeless. There are some counterexamples. So you really have to look at case by case what you're doing. So that's a very very difficult problem in some cases. Okay, so I'm not going to spend more details on that. I'm going to go to the next step in order to do coarse graining. Once you have the equation for the particle distribution function, you would like to sort of coarse grain it. So basically what you're doing is you're averaging over velocities. So you construct a local particle density or local mean velocity. And from the equation for F, you hope to get equations for these quantities, right? And one of the key features that is in classical physics is used to get these equations are conservation. So when you have particle interactions in a gas, when you have collisions, then you have a conservation of momentum. So imagine two particles colliding and getting after collision new velocities. Then what you have here, for instance, is that the sum of the velocities, I assume equal masses, the sum of the velocities after the collision is the sum of the velocities before the collision. So that's momentum conservation. You have also energy conservation. You have angular momentum conservation. You have these conservations, and these actually are key to obtaining macroscopic equations for these quantities, right? However, in the case of systems of, say, a little bit more disturbing agents, like for instance vehicles on the road, you don't have momentum conservation. Think of two vehicles. So there's a slow vehicle and the fast vehicle that gets stuck behind the slow vehicle. At some point, it sees that there is an opportunity for him to take over. So to pass, so in fact it passes, and then after the interaction, so you can see this as a collision, not a real collision, but an interaction. And after the interaction, this vehicle goes faster than before. This vehicle has the same velocity. If you sum the velocities, then you don't have the sum of the velocities here is larger than here. So you don't have momentum conservation, you don't have energy conservation. The reason is that, of course, these particles, of course, the physics is not violated because they are taking this energy and this momentum from, burning, gas, right? So this is basically, this feature arises because you're looking at a subsystem. You're not taking into account the gas in the tank, right? So, but you can't, right? In general, you can't really extend your system up to getting a closed system because it's too complicated. You have to live with open systems. And in this open system, you don't have these conservations. So, so it looks like it's hopeless to get macroscopic equations. Actually, this is going to be the subject of the second talk, we managed actually to find that there are kinds of weaker conservation relations. We call that generalized collision invariant, that allow us actually to coarse grain the kinetic equations and get macroscopic equations. So for instance, in the case of the Vicsek model, one of the goals of the second talk is to prove that we are able to show that the density, the local density and the local orientation of the velocity, so here u is not the velocities of the particles, but the velocity orientation. Remember, the particles have constant velocity in the Vicsek model. They are sort of self-propelled like sperm. So there is no such thing as conservation of momentum. However, you can write an equation for the velocity orientation. So that's a unit vector. And that velocity orientation you can show that it satisfies this evolution equation, which is not a conservation relation. It's not a conservation equation because you have you know, you have this non-conservative product here. I will come back to that later. So there is some hope. However, so this system we refer to as the self-organized hydrodynamics. It looks like a kind of a hydrodynamic system that has some special features. So it's this self-organized hydrodynamics. This is the work I did with my former student, Sébastien Moch, who is now in Arizona. So one of the goals of the talk will be to show how we get this system. Just to show you that this actually works, this is a simulation of the Vicsek model with a large number of particles. This is how, you know, the density in color-coded and velocity with arrows, which is directly obtained from these particle systems. And here, actually, these two are the same. It's not the same time. This is why it's different. Actually, not so different. So these two are the same. And this is actually this coarse-grained model, the self-organized hydrodynamics. And you see that there is actually very good agreement. We actually went a little bit further. So, you know, the original model is a stochastic model, so you can't really rely on a single realization of the system in order to get a comparison, a fair comparison. So here in this case, we performed actually an average over 50 realization or something like that, and compared with one realization of this macro system. I don't know if I can reset the video again. Let me try. It's kind of strange, I cannot. So yes, so as you see, there is an excellent agreement. Actually, the microscopic model, the stochastic dynamics, actually produces a you know, stronger clusters. There is a little bit of diffusion smoothing in the macroscopic dynamics, but that's actually quite good, and we have documented the way the two models approximate each other. So it's really proven. Actually, you can see in physics, there is a physics literature about the coarse graining of the Vicsek model, which is not based on a rigorous mathematical analysis, but rather more on, say, heuristic arguments. And they come up with models which are not very far from this one, but not exactly the same. There are significant differences. The first one who did this coarse graining in the physics literature, these two authors, John Toner from Oregon and Yuhai Tu. And yeah, I've been discussing with John Toner quite often about these differences. I think the physics approach is very interesting, is very intuitive, but it's best to have a rigorous, you know, mathematical procedure actually to assess the what you get in the end, right? So now let me just show you a certain number of current projects, also to give you some perspective. So here I like to view actually this. I like to work in collaboration with biologists and actually other fields of science. And when they propose me to work on something, the first thing I try to imagine is that I'm trying to build a model using actually like a bit like a construction game. So I have some, you know, some bricks, and I have some construction rules. So the bricks are the types of particles I'm using in the model, and the construction rules are the interaction. And I'm trying to find how, what are the right bricks and what are the right construction rules. So typically here in this model, which is intended actually to sort of propose a possible scenario for the formation of these lobules in the adipose tissue, we imagine that we have cells that are spheres actually in 2D. These are disks, and the fibers that surround the cells are modeled as line segments that can cross-link, right? And so we have put in cross-link and unlinking rules, and also some alignment rules between the fibers. And you have to play with the parameters, and so with some parameters you get structures that are actually quite similar to what you get here. So they are not quantitatively really similar, because as you can see there are less cells, and maybe the fibers are not exactly the same. But we are not really interested in quantitative agreement, because if you want to get quantitative agreement, then you are forced to pour in more and more, ingredients in the system, and in the end, you you have such a complicated system, you cannot really work with it. And rather what we're interested in looking at trends. So what happens when such parameter is modified? So for instance, is decreased or increased, right? And so for instance, if the alignment between fibers is decreased, how do we modify the structure here? And does it relate to some pathologies that we can observe in the tissue? So we can actually suggest the biologists some of the mechanism by which some pathologies can appear, right? And then, of course, the biologist, with this suggestion, has to verify it experimentally by using drugs and experiments and so on. So it's more like using these tools as a kind of a way to explore possible mechanisms, right? So here the interaction, I forgot to say the interaction rules are cell-cell volume exclusion. So we actually here implement a hardcore volume exclusion. This is not a repulsion force. This is really a constraint, a hard constraint. Then repulsion between cells and fibers to model the containment of the cells by the fibers, which is one of the roles of the fibers. Cross-linking and unlinking and alignment of fibers, as I said, right? So that was a work that was the subject of the thesis of Diane Perichat, who, by the way, is looking for jobs. So if you're interested, ask me. Okay, so that was the advertisement section. Yeah, so there's another thing that we've done in the same spirit is actually looking at blood capillary formation. So here we have fluid which is supposed to model the flow of blood or interstitial fluid, which is modeled by using the Darcy's equation of in porous media. Then we have oxygen, which is in red here, which is satisfies the convection diffusion equation. And then we have capillaries, which are these blue line segments. And the rules are transport of oxygen by the blood, capillary creation of deletion in response to oxygen concentration, and the fact that when you have a capillary, and that's the important feedback mechanism, that it increases the hydraulic conductivity of the tissue. So that means that if you have a pressure difference, when you have a pressure difference between two ends of the capillary, then the blood has of course, has a higher facility to flow in the direction of the capillary. So this basically, the capillaries just locally modify the properties of the tissue. And you see that there is a very nice branching structure that occurs. And here comes a problem, actually, that we'd like to analyze these branching structures, and we're lacking some tools, right, to do that. So how do we analyze this branching structure, the angles, the width of the channels, and so on? So we really have to develop statistical tools to do that, skeletonize this network, trying to get statistics of branch length and branching angles, and so on and so forth, in order to explore the influence of the parameters of the model onto the type of structure that you get, right? So this is actually a lot of work, because even if the model is simple, you have a lot of parameters, and you have to find the good ranges of parameters and how to play with them, and that's a lot of work. The simulations are also taking a little bit of time. The last thing I'd like to mention before I get to the more mathematical part is this last work where we try to actually analyze alignment, but not only a velocity alignment, but the alignment of the whole body attitude of the subject. So this is basically trying to think that these two geese would like to not only align the direction of motion, but their whole body attitude, right? So body attitude is described by a frame. So you're going to describe a generic frame by taking a reference frame and a rotation matrix from this reference frame to this generic frame. So here, for instance, you have an expression of, okay, so typically if you think that this is the reference frame, then you have a rotation to get from here to here. Now, if you think that these are two bodies that try to align, you see that the two frames here are unaligned, and then here they are aligned. So So essentially, that's playing alignment on rotation matrices, right? So what this gives is this. So we implemented this. And so the color code is that we think of these, these birds as tetrahedra. So on one side of the tetrahedron is blue, the other one is red, and the third one is green, and the back is white, okay? And so what you see is that after very quickly there are clusters that form, a little bit like in the Vicsek model, but what you can see also is that these clusters have the same color, which means that they are all oriented, the body is oriented in the same direction. However, between different colors, that means that the body frame is oriented in different ways. Okay, so we'd like, in some sense, to analyze this. And one of the questions that we had in mind is that, does that produce a dynamics that's different from the Vicsek model, or is this just, Vicsek alignment plus something, rotation of the body frame? Right? So is there a true interaction between the dynamics of the body frame and the decision that the particle take about the direction of motion? And so it's not easy to answer this question on these simulations. So what we again used is coarse graining. Okay, so what we did is coarse graining, and that's a joint work with Amit Frouvel and my postdoc Sarah Merino, and Amit is in Paris Dauphine. And so we found that we have an equation, so rho is the density of agents, but lambda is the mean body attitude represented by your rotation. So you have to think of lambda as a rotation field, so every point in space and time has a given rotation, and this rotation evolves according to this equation, which is a little bit clumsy, but essentially what you see here, if you're looking, if you are trained, I am, you are not, obviously, but let me give you the answer. If you're just looking at this block here, forget all these two terms, then you get essentially a glorified version of the Vicsek model. But these two additional terms, and these R and delta, are actually kind of curl and divergence of this rotation field in a way that I'm not going to explain you because it would be a bit technical. But these are actually showing that there is a true, the true effect of the local body orientation of the agents into the direction they are taking. So this model is not just Vicsek plus something else, it's really a different model, right? So this is typical of something that we just, and actually we have, since this work, we have done a quaternion version of this because rotation matrices are also can be encoded in a unit quaternion, right? And so quaternion is a four-dimensional vector. So it's a bit nicer because instead of having an object in a group in a non-obvious group, rather than you get an object which is a unit quaternion, which is basically an object which is on a unit sphere in R4. So it's a little bit simpler, but essentially it carries the same information. So this is just a kind of overview of different aspects. Just let me give you a conclusion of this first part. So emergence is a property of systems that develop patterns on scales larger than those of the individual components, as we have seen. So emergence can be viewed as a phase transition because you have a transition, say, from disorder to order when you have, in a very narrow range of parameters of parameter space. Actually, kinetic theory, and I would say the tools that allow us to get mean field models is method of choice to derive models of emergent behavior. So it's actually in line of Hilbert's sixth problem. And so what I want to actually also highlight is that emergence requires developing concepts beyond the state of the art. So we had seen the problem of the propagation of chaos and the problem of conservation of momentum, and there are other problems that I did not mention here, which require really the development of new concepts. So it's a very fascinating problem for area for mathematicians because you really have to develop a new imagination of the problems. Yeah, I have been collaborating with many people. I'm not going to list all of them, and also I had a chance to get money from here and there. Money is, you know, le nerf de la guerre, as we say in French, so it's important to acknowledge that. Just a comment that these slides are just an excerpt of my inaugural talk at Imperial College. You can find the whole talk online on Imperial College YouTube channel, and there is a link on my webpage. But I would say the most scientific part of this is what I've just said. So I'm done with this first part, and if you, I would like to take some questions before I move to the next part. I don't know if you have any questions. Yeah?
You spend a lot of time discussing the role of inertia in living systems, where you compare momenta of particles before and after collisions. But is that does that play an important role? In typical examples, is there not a path or a fluid around which completely damps the inertia?
It's not really inertia. I mean, it's really conservation. The fact that there is a mass involved is coincidental. I think that's not the problem of inertia or not. It's really the problem that essentially, so when you're in equilibrium, when you are close to equilibrium, when you're at a local thermodynamical equilibrium, a local thermodynamical equilibrium has some free parameters, right? Like the density, the mean. So for in the gas, in the case of the gas, the local thermodynamical equilibrium is a Maxwellian. It has free parameters: the density, the mean velocity, and the temperature. So this is a list of 5 parameters because the velocity is a vector in R3. And so in order to determine their equations, you need 5 relations, and they are provided by the 5 conservation that you have: conservation of mass, conservation of momentum, and conservation of energy. So you have a good match between the number of conservation and the number of free parameters in your local thermodynamical equilibrium. In the case, and I'm going to develop that in the case of the Vicsek, you have local thermodynamical equilibria which have a certain number of parameters, but you have less conservation equations. So you're missing information in order to determine the evolution of some of the parameters. So that's the difficulty. So yeah. So I mean, the fact that it's related to mass is coincidental. Now, what you say about the role of the fluid is actually important. There is, in fact, in this, physicists have a common way to classify these collective dynamic systems in active matter. So they call dry active matter when you have particles that are moving independently of the surrounding medium. So take, birds. Birds fly. They do not take into account the fact that they fly in air, in a gas. Okay, they just take into account the fact that they interact with each other and they are self-propelled. So this is a model of dry active matter. On the other hand, you have soft active matter where typically this is the case of the sperm, where the sperm cells are embedded in a fluid. And of course, the fluid mediates the interactions between the cells. So there is hardcore direct interactions, but also long-range interactions which are mediated by the fluid. So there is a different class of models that deal with these soft active matter models, but to some extent, the problems that arise in both kinds of systems are very similar. And the mathematical problems are those which are listed here.